Today I'll be talking about label-free high-throughput image cytometry, a little bit about the company I work for first. I'm actually a field application engineer. Before COVID, I used to travel all around the world working on high-speed imaging applications. So today I want to talk about again image cytometry and a little bit of background on the webinar. I'm going to just talk about traditional cell cytometry and how it contrasts with image cytometry and what are some of the benefits and drawbacks and so on. Then I'm going to talk a little bit about the key challenges of high-speed imaging and why it's important to understand the challenges and also the ways to overcome them. I'm going to show you a couple examples from literature to give you an idea of how it works. And then I'm going to give you my perspective on where I think the field is heading. And this will deal with what are known as machine vision cameras and a real-time image analysis and feedback. This is kind of more of a traditional look at flow cytometer. Basically what you have is you have forward scattered light collected, and then you'll have basically multiple fluorescent channels that the system will analyze and be able to tell you, what particular fluorophore is in your sample, and thus what kind of chemical and biological species exist in your sample. Now, image cytometry it's actually a little bit more simple, at least in the experimental point of view. And flow cytometry is basically, or image cytometry is a technique used to capture the physical and chemical characteristics of a population of cells or particles with digital images. Okay, and the way it works, on the right you see it has a high-speed camera, a microscope objective, a long working distance microscope is fine, or a traditional microscope is okay, a microfluidic channel just like what you would have with the flow cytometer, single cells or single file micro objects. And then the way we do high throughput imaging and image cytometry is we use a intense LED light and we shine it from the back of the sample. So it's like traditional light microscopy. And this technique is also used for cell counting, sorting, identification, detecting microorganisms, looking for biomarkers it could be used for, diagnosis of health disorders, and real-time analysis and feedback. So this is a typical example of a high-speed video of a cell going ultra fast. So what can be quantified from this? Size, shape, granularity, the rotation of that cell. You get a 3D view of it. People are working on determining elasticity of that as it travels through, the opacity, and you can even look into, with high quality, high speed imaging, you can even look into the organelles and see what the inner side of the cell actually looks like. So here's an example of real-time image cytometry where we're looking at, in this case, it's a model system looking at 25 micron polystyrene beads traveling through a microchannel. And what I'm going to show you is a video captured with 2048 by 72 pixels. Okay, that's the resolution. And it's actually occurring at 3,300 frames per second. So what the software is actually doing is not only automatically detecting when the object comes into the field of view, but it's also getting the real-time position and velocity of that object. That's all for today, guys. I hope you liked the talk. If you have any questions, please don't hesitate to reach me. My email is listed here. Please have a great day and be safe. Thanks.